Hey kids, welcome to Doing Nothing Academy. My name's Bernard, because self-inserts are not cool. Today we're going to talk about my favorite type of jeans, Levi's. Nah, I'm just pulling your leg. We're going to talk about DNA, or if you're an elitist, deoxyribonucleic acid. DNA is like Cheerios, you're going to need it to have a good morning. No complex life can exist without it. DNA is made of two connected strands. This weird ladder-looking thing is called a double helix. It's unlike RNA, which isn't quite all there. I mean, look at him. He's basically half a person. The first person to isolate nucleic acid was Johannes Friedrich Meister. At one point, he even worked with Adolf Strechter in 1865. The big discovery for us, though, is the double helix shape of DNA. This was done by James Watson and Francis Crick. They're quite the characters, but Watson is especially interesting. This video isn't about him, so I'll suffice to say he had quite the racy history. Another person who deserves credit is Rosalind Franklin. Her research was yoinked by Watson and Crick to make their big Nobel-winning discovery. She received no formal credit for the discovery because she went and died before she got the prize. Buckle up, kiddos. We're going to have to use some big words now. DNA is what's known as a polynucleotide. For the crayon crunchers in the back, that means it's made up of a monomeric unit called a nucleotide. Nucleotides are complex little goofballs. They have all the bits that make DNA possible the way we know it. Nucleotides are composed of the sugar deoxyribose, a phosphate group, and one of the 4 nitrogenous bases. The sugar and phosphate create the backbone to which the nitrogenous bases can bind and make the magic happen. What's the magic, you may ask? Well, listen close. Each of the bases is special in its own way. We've got 4 types to work with: adenine, thymine, cytosine, and guanine. We don't mess with uracil. He's from a bad part of town. These magical little hexagons are the rungs that connect the sugar-phosphate strands together. The bases bind to each other through hydrogen bonding, a form of bond almost as weak as half the class's work ethic. The bases are segregated into 2 groups, the purines and the pyrimidines. The purines have a double ring structure, and the pyrimidines have a single ring structure. They only pair to the opposite type of base. This is called complementary pairing. They're loyal. They only bond to one person in their life. Unlike Karen, you cheating. Now these bases are ordered to create genetic code. The sequence gets spread by enzymes in your body to create proteins that make you a person. The code isn't close to perfect, though. In humans, more than 98% of DNA is effectively useless, similar to fad diets. Let's go back to the structure of DNA. Deoxyribose is a 5-carbon sugar, with each carbon numbered 1 through 5. The DNA strands are anti-parallel to each other. The orientation of DNA is shown by the 5 and 3 prime ends. Think of the backbone as a road, where the orientation of the deoxyribose sugar shows which way you need to drive. Enzymes use this in the replication process DNA undergoes sometimes. The sugar and phosphate are alternating in the backbone. These bonds are strong, stable, and most importantly, aesthetically pleasing. DNA doesn't just stay in worthless messy coils everywhere, though. It forms a tight package with tons of DNA called a chromosome. Now these are some tight homies. The chromy homies. They're the blueprints for you, your homies, and everyone else. Alright kids, that's all the time we have for today. Everything we learned about today is what creates the magic, stable, self-replicating little molecule that we all know and love, because it tells us to.